we are left with the two topics. We are left with the two topics in this chapter. First is called as the growth and multiplication in bacteria, and second is called as the culture media, means basically the identification of bacteria, how we will identify the bacteria in the lab. This two is left. So today we will cover the growth and multiplication, and from tomorrow class we will cover the identification of bacteria. Got it? No problem. So till now we understood that apart from us, the human beings, there are other organisms which is living on this planet, and bacteria is the most abundant living organism. Among all the organisms which is present in this planet, bacteria are the most abundant. Okay, means largely they are found as compared to the other organism number wise. Okay, but if we talking about the size wise, then it is a tiny, it's a very small. Now although it is a small, but as we discussed that bacteria do need nutrition for their survival. Bacteria do need nutrition for their survival. So after getting the nutrition, bacteria will grow as us. After getting the nutrition, we will grow. In the same way, by getting the nutrition, bacteria also shows the growth. So for growth, the two things is required. For the growth, how much thing is required? Two things is required. The one is called as the cell division. Okay, first one is called as cell replication, and then cell division. And again that cell division is followed by the cell differentiation. For growth of a bacteria, the thing is needed is first they will replicate and they will divide. That is the main basis of cell division. Okay, or we can say the growth. Fine. Now if we are talking about the growth, then we will define growth is in the two way. First one is increase in the cell mass. Increase in the cell mass, or either increase in the cell number. Increase in the cell number. That is called as the growth. That is called as the growth. Growth means increase in the cell number or increase in the cell mass. Either if we take the example, if this is a cell, then if we labeled it that this cells, it shows the growth. In the two way we can label. First, if their number increase, or if the size increase. If the size increase. So growth is defined as increase in the cell number or increase in the cell mass. Fine. Now, so as after getting the nutritions, there are many nutritions we already discussed, that some bacteria truly you can say based on getting the nutrition from the dead things. Some bacteria are truly dependent upon getting the energy from sunlight, that is called as the photoautotropic bacteria. Then some bacteria is dependent upon some other organism, that is called as the parasitic bacteria. Getting the points? There are different mode of getting the nutrition. So once bacteria get the nutrition, it will increase its size. Fine. And then it will divide. So which is the preferable mode of bacterial division or multiplication? Come first. Which is the preferable, first listen the question, which is the preferable mode for the bacteria to produce the another bacteria, or you can say to give the birth of the another bacteria? Which is the preferable mode? We discussed reproduction in bacteria. It is a binary fission. So as I mentioned earlier, that binary fission is the normal mode of reproduction in bacteria. But apart from the binary division, sometimes during the certain circumstances, like in unfavorable situation, bacteria do perform the another type called as the sporulation, and one type is the budding. Okay, clear? So generally bacteria through the binary fission, it will multiply into the two another bacteria. So this is what called as the growth. This is called as the growth and multiplication. Clear? Now, for one bacteria, they need certain time to replicate. They need certain time to replicate. As a human, we cannot give the birth in a one day, isn't it? It takes 9 months plus minus 7 days, called as the gestation period. In the same thing, bacteria do have, that is called as the generational period. That is called as the generational period. Write it down. That is called as the generational period. Now, what is the generational period? It is basically population doubling time. It is the population doubling time. Means from one bacteria, it will multiply and form the two bacteria. So the time taken to multiply from 1 to 2, this much time period is called as the generation time. This is called as the generation time. You can write down. E. coli shows the generation time 20 minutes. Okay? E. coli need 20 minutes to double their population. E. coli needs 20% to double their population. Fine? Retain? Yes? E. coli needs 20 minutes to double their population. And this time period is called as generational time period. This is called as the generational time period, or also called as generation. Simply generation time period or generation time. Clear? Yes? Now this is the interesting thing. Why they are more in number, okay? Or why after we are basically bombarding the bacteria with the antibiotics, okay? Like there are so many things. Vaccines is also one of the things by which we are basically killing the bacteria. But still they are present in us, okay? They are present in other organisms. They are present in the dead, decaying things. Are you getting the thing? Are you getting my points? Yes? Just because they have the very shorter time to double their population. They have very shorter time to double their population. That's why they are more in number. And how much we are killing, still they are in their existence. Still they are in their existence. कितना भी हम लोग उसको मारते हैं, फिर भी अभी तक वो प्रेजेंट दुनिया में है. जस्ट बिकॉज कि इसको अपना पॉपुलेशन को डबल करने के लिए मतलब 20 मिनट ओनली लगता है. इट टेक्स ओनली 20 मिनट. फाइन. बट अगर हम लोगों के साथ अगर किसी का मान के चलो कोई दूसरा ऑर्गेनिज्म आ गया विच इज सुपीरियर टू अस, yet there is no any superior organism. We are the only topmost organism. But अगर मान के चलो आ जाएगा हम लोगों से superior and अगर वो हम लोगों को मारेगा तो हम लोग तो ऐसे ही मर जाएंगे, क्योंकि हम लोगों को तो 9 महीना लगेगा आएगा जनरेशन को देने के लिए, जन्म देने के लिए हम लोगों को टाइम लगेगा. तो हम लोग का पॉपुलेशन शॉर्ट जल्दी खत्म हो जाएगा. लेकिन इफ यू टॉकिंग अबाउट द बैक्टीरिया, if you are killing, there are already n number of bacteria is present. You are start to killing but through the antibiotics. Okay? बैक्टीरिया मर रहे हैं, but again तुम 20 मिनट तक बैक्टीरिया को मारोगे तब तक दूसरा बैक्टीरिया ग्रो कर जाएगा. Getting the points? So it's the generation time is shorter. That's why they are more abundant. The first thing. Now इस 20 मिनट में बैक्टीरिया क्या करता है? What they will do in these 20 minutes. 20 minutes for E. coli. We are taking now the example, and through that E. coli example, we'll understand what things actually happening in these 20 minutes and how it grows. ये 20 मिनट में बैक्टीरिया, as I said, that ये grow करता है, fine. But it is not something कि 20 minutes takes time to pass. No, it's already 20 minutes. 60 seconds pass, then again 60 seconds pass, again 60 seconds pass. one minute pass, then it is the 20 minutes. So इस 20 minutes के अंदर में बैक्टीरिया के जो डिवीजन होते हैं उसमें कुछ चीजें होती है. Now you cannot see this growth pattern directly by looking at the bacteria, okay, in the blood sample or anywhere. You have to take the bacteria sample from you can say many sources. In the saliva, there is a bacteria is present, okay? In the urine, bacteria is present. In the blood, bacteria is present. Are you getting my points? In the pus, bacteria is present. So we will take the bacteria, okay? And then we will inoculate. Inoculate means basically we will transfer from sample. After making the samples, you can say clean, we will take the bacteria and we will put into the where? Yeah, media, the nutrition media, fine. And we will culture. Culture means we will provide the adequate, you can say supporting nutrients. See, each bacteria need a different type of nutrition. Different type of nutrition. You can take you are Bihari, you most prefer like rice in your meal, like in the lunch time, and at the dinner you need chapati or roti, fine. South Indians, they follow the different pattern. Are you getting my point? Still the nutrition is needed for all the organisms, but there is a choice. The bacteria also have the choice, although they are very small, but uske paas bhi uska choice hai. So according to the choice, according to the requirement, we will provide the nutrition to the bacteria in the culture plate or culture media, fine. We will given all, now bacteria will grow and forms the colonies, that colonies is visible by the eyes, that colonies is visible through the naked eye, fine. You cannot see the single bacterium through your naked eye because they are too small, they are beyond to the human eyes resolution, so you cannot see. But what you see is their colonies. What you see is the colonies, fine. And this colonies is like, suppose, okay, I rub this thing. It is clear, no? Fine. So through the diagram, we'll learn what is actually happening with the bacteria in this 20 minutes. So thing is, we take in the sample. Yeah, come fast. Questions low, take the questions and start writing. It's only 10 mark. Fine. Yes. So from the human, you can say the human blood. Okay. From the human blood, we take in the sample. Okay, we taking the sample and we put it into the first test tube. Okay, we put it into the test tube. Now, from this test tube, we will inoculate. What we will do? Inoculation. Inoculation. First step is what? Sample collection. Sample collection. Collected. Now inoculated where? Into the media plate. Into the media plate. Fine. Successfully we are now. This media plate, no, we will have the detailed lecture tomorrow, but for a while you just understand. It basically gives the bacteria their preferable nutrient, their preferable or their according to their choice. We are basically preparing this culture media. Fine. Now it can be the liquid, it can be the solid, or it can be the semi-solid. So after giving all the nutrients to this is the ready. Now these samples we are giving here, and after some time, okay, bacteria like come here. This is not visible through the naked eye yet, but after you can say some time period, you will see the bacterial colonies. Now, these colonies are visible through your eyes. Getting the points? And not Not understood? Fine. So first thing is, from the blood we taking the sample, okay, taking the basically the blood, and that blood contain basically the any bacteria, okay. Now this bacteria we are putting into the test tube, okay, putting into the test tube. After putting into the test tube, we will take the bacteria and again put into the nutrition media, or you can say nutrition culture media, fine. And according to the bacterial nature, here what we are going to identify is bacteria is aerobic or anaerobic. We will come to know from this test tube, differential test tube, and then we will put into the culture media, okay. So whatever nutrition is required, this is already present in this culture media. Now this, after getting the nutritions, if it is a E. coli, if it is an example of E. coli, then E. coli will take 20 minutes to double their population. 20 मिनट उसको लगता है अपने पॉपुलेशन को डबल करने के लिए, फाइन. आफ्टर द 20 मिनट्स जो बैक्टीरिया को तुम नहीं देख पा रहे थे, इट्स वेरी स्मॉल, नाउ इट्स फॉर्म द कॉलोनीज. व्हाट इज फॉर्म्स? इट्स फॉर्म द कॉलोनीज. Now this colonies is visible through human eyes. इसे तुम अपने आंखों से देख सकते हो, ऐसे उठा के तुम देख सकते हो. Fine? Now, in this 20 minutes, basically 4 things happens with the bacteria. If you're talking about the bacterial growth pattern, that like for example in the humans, कैसे development होता है? पहले हम लोग गुरक के चलते हैं, like we are crawling, okay fine, then we stand on our own feet, then we start, you can say walking, then running, jumping, climbing. It's a slow progress. It is a slow progress. Fine. बैक्टीरिया के साथ भी सेम थिंग्स होता है. Are you getting my point? बैक्टीरिया के साथ भी सेम थिंग होता है. So इस 20 मिनट में मैं एक्चुअली व्हाट आई सेड इज द 4 चीजें बैक्टीरिया के साथ मेनली होते हैं. 4 चीजें. फाइन. नाउ स्टार्टिंग विद द फर्स्ट, it is called as the lag phase. It is called as the lag phase. Fine. Then the another thing which is happening with the bacteria is the very good log phase. Yes, the next thing, and then phase of decline. Phase of decline. ये 4 चीजें, this 4 thing is happening with the bacteria in this 20 minutes. Fine. Now start.